Good evening, everybody. My name's Andrew Adams. I'm a second-year student working on a Master's of Biology focused on stem cell research here at Sacramento State University. Tonight, I want to welcome you to our Topics of Regenerative Medicine seminar series. Tonight, Dr. Richard Bouillet will speak to our community about the translational applications from canine to human patients. This talk is the last of our four-part series intended to provide information on the advancements in stem cell and medical research to the general population, including our undergrad students, our graduate students, and professionals and others within the community. This series is co-sponsored by the Center for STEM Excellence, which promotes the advancement of science, technology, engineering, and mathematics discipline, as well as CIRM, the California Institute for Regenerative Medicine, which provides our program with funding and allows students to collaborate with researchers at UC Davis, investigating topics such as neuronic diseases, bone and wound healing and repair, as well as diabetes and cancer treatments. We're fortunate tonight to have with to have you all with us this evening. All of our past, current, and future speakers' seminars can be viewed on the Sac State STEM website. We are currently recruiting applications for our next year's cohort of students. And if you are interested in the program, please contact or come speak with Dr. Carter down here in the front row later this evening. It is my pleasure to introduce our speaker today, Dr. Richard Bouillet, a professor at UC Davis's School of Veterinary Medicine. He received his PhD at the University of Colorado, Denver, along with his Doctor of Veterinary Medicine at Colorado State University, Fort Collins. Dr. Bouillet has received numerous awards for his work, and he is also the founder of REGEN Laboratories, LLC. Please join me in welcoming our honored guest.
Thank you, Andrew. I want to express my sincere appreciation to be invited here to give this presentation. I always enjoy talking about stem cells in dogs. But what I enjoy most is giving a talk in the same time zone I live in, and also the fact that I didn't have to get up at 3:30 this morning to catch a 7 a.m. out of SFO. So it's nice to be among friends in a local atmosphere. And thank you all for coming and missing what I hope is the last game of the San Francisco Giants. At least that's what we're hoping for. So, okay, what I want to talk about is I want to talk about the use of dogs and dog models. And I loathe the term models when I'm talking about dog patients, because to me they're people and they're patients, not models. But when you're talking to NIH researchers, you have to call them models because they just don't see, I guess they don't have pets or something. But how many in this group have a dog at home? Excellent, about 50%. That's very typical. It may serve good notice to you that you can get treatments in your dogs that you can't get in yourself right now. Interesting. So, okay, with that, let me talk about my generation, because I see it's a younger crowd. My generation used to worry about how many angels danced on the head of a pin. That was the big philosophical question. Your generation is how many stem cells fit on the tip of a pin. So as you can see right here, and this is the picture of the company that I founded in Davis called REGEN Laboratories. Pamela, will you stand up? If you have any dogs you're interested in enrolling in our study, this is our clinical trials coordinator, Pamela Rossman. She'll be the person you talk to and let you know what we're interested in terms of screening. But with that, yeah, she's the one that does emails, so I'm too busy pipetting. Anyway, most of you, this goes back, and you know, that's how you can tell you're getting old, is when you start talking about history rather than what you did, rather than what you're going to do. But in point of fact, it's important that you keep a concept of where you are in the time course of events. In modern therapy started probably at the turn of the last century, in about 1900s, 1920s, with Fleming discovering penicillin. That was the first really modern drug that we had that stood the test of time. And each wave of therapy has cascaded upon it. We discovered receptors, and we discovered receptor blockers, and we discovered different things. But your generation in this area right here is dealing with chronic degenerative diseases, losses of many cell types, and with the exception of something like diabetes, where you replace the insulin molecule, if you're trying to replace these cells, it's going to be a good deal more complicated than previously. Finding a single chemical that kills a bug and doesn't kill mammalian cells is relatively trivial in comparison, although it was a big step forward. And what happens is, once you find that magical cure, the other thing we're struggling with as a society is, how do you get it from discovery phase right there out to in the clinics with an improved clinical application? Transversing this spectrum is about $800 million and takes 10 to 20 years. So don't plan on buying a new Corvette as soon as you make a discovery right here, because you're probably not going to be able to afford it, and for a long, long time. That's a problem. Okay, it's not a problem because of the money. It's a problem because we have dying patients. We have patients that are suffering. Should they have to wait 20 years for a treatment? And the answer is, I don't think so. So question is, how can we speed this up? How can we make it more effective, more work for our population, at the same time providing safety? Because safety is the first and foremost consideration that you have. And so that's what this talk is about, and you can see the credibility gap right there. I sail under the San Francisco Bridge frequently, and I haven't seen a car go down yet, but it is a concern. Okay. One of the reasons that there is so much distance in here, you make a fundamental discovery in mice, and I'm going to come out across as a mouse hater, and I'm not. I like mice. Actually, I don't like mice, but they have a use in the biomedical research field, but I think we have gone a little too far. And when I say that, this is an example for treatment of ALS, amyotrophic lateral sclerosis, better known as Lou Gehrig's disease, terminal disease, horrible disease, ascending paralysis, where you get up each day and you're a little less able to do something than you could the day before. Horrible disease. And what they did is they took a drug called Celebrex. Celebrex is a non-steroidal anti-inflammatory drug, in the sort of like aspirin, only it's a new improved aspirin. Somebody had tested it in a mutant mouse, an SOD1. SOD1 is superoxide dismutase. It's an enzyme that has been shown to be the mutation in familial ALS. So you have humans that have familial ALS. That's about 5% of the ALS cases. They have a mutation, the SOD1. So you pull that mutation out, or you take a mouse, you mutate a mouse with an SOD1 mutation, and you say, aha, this mouse has ALS. Okay? So then what you do, now that you've made this mouse, is you start testing it. So you test it with a drug called Celebrex. And what you do is measure, this is called the Kaplan-Meier curve. This is survival here, this is time here, and you can see there's a difference between the treated and the untreated mice. I got it. Cool. I've just shown that Celebrex treats ALS, right? Anybody want to invest stock in the company? Okay. Well, this was about $300,000 to do that. This was about $10 million. And because Celebrex was an approved drug, you just go to the pharmacy and get it. So I just round up 100, actually it was 300 patients, or 200 ALS patients, and I measure, over time, I measure their, it's called the ALS score. That's an index of how well they're doing, and an integrated formula that they use to calculate response. There's no difference in those lines. Whoa, wait a minute, these mice work. Didn't work here. Well, that was okay. Then we tried it again with a drug called minocycline. That's a tetracycline-like molecule. Again, you had untreated here, treated here, difference in survivability. So, and again, this is drug approved in humans. Great, we can just call up, dial up, go to the pharmacy, get some drug, find an ALS patient, put it in. Again, no difference there. And these aren't lousy labs. These are good labs, peer-reviewed publications. So, I mean, Lancet, you don't get better than Lancet, you don't get better than Nature. And so what's going on here? Interesting, isn't it? And yet, and a third time, they tried it with lithium. Lithium's an antidepressant drug with mice. Again, you saw increased survival. And what they actually halted the human trial with lithium in humans because they actually had adverse reactions. It was dangerous to the patients. It wasn't just not doing anything to the patients. So what's going on here? Well, right now, there are no really good models of ALS. And stem cells, I remember from San Diego, the stem cell group had a new approach to ALS, and I read the flyer on it, and it said, this new approach is so good, we've tested it in 11 different ALS models, and it works in all of them. And you know what? The FDA finally said there are no good models of ALS, and there's a clinical trial being done in Mayo right now in Rochester, where they just used a dose escalation. They just said, okay, there's no good mouse models we really trust, so what we'll do is start off at a low dose, and each time you can increase a little bit more dose till you get a thing, because the mice just don't project onto what humans are doing. This was probably the final nail in the coffin, so to speak, in terms of doing mice studies. This was a study done by investigators at Brigham and Women's in Boston and Stanford, where they looked at the genetic expression in mice at using either burn, trauma, or endotoxemia. These are 3 different insults to the human animal and the mouse animal. And if you had similar responses, these heat maps would look exactly the same. They would look like twin pictures. They're not even close. Okay, so you get different genetic responses. This is one looking at time course, and the time course is completely different. So you need to be aware of your model and what you can say about your model and when it's restricted. But the statement that almost everybody clinician says is you can't go from mouse to man. You need something else in between. And so what we're talking about is, we started looking at the idea of if you can't use that, what can you use? And what turns out that this is humans, if you look at the 11 major diseases of humans, it turns out dogs have at least 9 of them, and it's not clear whether they get strokes or Alzheimer's disease. It's probably not the formal Alzheimer's disease, but I can tell you geriatric dogs that are really old do get senile. I can tell you dogs do have thromboembolic events, but they're generally not as a stroke in terms of what humans see in stroke. What else do we know about dogs? We also know that dogs, if I ask everybody to conjure up a picture of a dog, each one of you would have an image of a dog that would be unique to you. But if I told you to conjure up an image of a boxer or a collie or something like that, what you would find is that you knew exactly what a collie looks like, what a boxer looks like. What there is in dogs is there's tremendous interbreed heterogeneity, but there's incredible intra-breed homogeneity. And because of this homogeneity, breeders have bred for coat color, whatever size, speed, but they also brought diseases with them that they didn't know about at the same time. These are spontaneously occurring mutations, but if I'm breeding a Dobie and I want a red coat color, and it happens to have a linked disease with it, and I breed for all red coat colors, pretty soon there's a lot of disease associated with that color. And that has happened in purebred dogs. Okay, and my hypothesis is, and suggestion is, that we can use these naturally occurring diseases in dogs to cross this valley of death, as it's called, because this valley of death is where a lot of compounds fall out before you get to clinical testing, that you can speed up the trial and save a lot of money, because this is $800 million to get across that. You can cut that down, you can do a good dog study for $300,000-$400,000 and generate probably better data than you can in all of the mouse studies that you've done. if you can find a dog with the same disease as your target disease. And I'll show you some diseases. What there is in the biomedical research community is very little appreciation of dog diseases and how close related they are in the diseases we share with our dogs. And that's what this whole thing's about. Can we use this to speed up the approval process? That's important, not necessarily for money, but in terms of human suffering. One other thing, most of you know what stem cells are. The ones I'll talk about tonight are the bone marrow stem cell, one other thing you need to be aware of is many of the diseases that are currently targets of biomedical research are middle-age or older diseases, okay? You know, the young people don't have diseases, but there's a lot of diseases go along with aging. Part of them can be related to MSCs. If you look, newborns have very high levels. Teenagers have 1 in 100,000. This is data from Arnold Kaplan's lab. When you get to 30, 1 to 50, by the time you get to 50 years of age, you're 1 in 400,000 stem cells per that. That probably has something to do with the onset of many of the adult onset diseases. If you think about it, your genes never change. But when you have a genetic disease, like ALS, like Huntington, something like that, you've had that same genetic makeup since you were born, but you don't see clinical signs till you're 30, 40, or 50, in the third, fourth, or fifth decade of life. So if there's something else going on, there's something maintaining the body, and then you run out of it, and it could be that the MSCs run out with aging. Okay, let me give you an example of an adult onset disease that is heritable and probably genetic, but it shows up. And it's a disease called idiopathic pulmonary fibrosis. Has anybody ever heard of it here? Okay, idiopathic pulmonary fibrosis, I was told by the people who do IPF research that it's second to breast cancer in terms of killing Americans. Marlon Brando died of it. Evel Knievel, if you remember Evel Knievel, the guy who was going to leap Grand Canyon on a motorcycle, died of it. It's a horrible disease, and what it is, and the idiopathic means, idio means cause unknown. Idiopathic means we don't know why it's there. Pulmonary, lungs. Fibrosis, fibrosis or scarring of the lungs. What happens is every time you breathe, you breathe in, your lungs expand. Every time you exhale, they constrict. Okay, now if you start adding scar tissue to those lungs, they don't expand as well, they don't constrict as well. What happens is you can't breathe from the inside. It's like being strangled over a long period of time. It's a horrible, horrible way to die. Turns out we don't know much about it. There's no treatment for it. And what happens is, ironically enough, what we know is that West Highland White Terriers, and most of you don't know what a West Highland White Terrier is. It's a little white dog. This is a West Highland White right there. And a low percentage of them spontaneously develop a disease very similar to IPF. If you look at the histology, it looks very fibrotic. There's scarring of the lung tissue. They eventually lose exercise tolerance. They eventually start gurgling. Fluid builds up in the lungs, and we pull the plug. We euthanize them. Can't do that with humans, even though they want you to do it. You can't do it to them, you know, for legal and ethical, and we won't go there because this isn't a religious talk. But what happens is, this is give you a history of some of the things we did. We started with our first dog, patient number 1 with IPF, a Westie on the East Coast. Okay. And we started with Westie on the East Coast. Lucky wasn't so lucky. We gave him 1 dose of mesenchymal stem cells. And in 3 weeks, he didn't get better. He was suffering sufficiently that we pulled the plug and euthanized him. Was that a failure or was that a success? Well, we usually start with more severely affected dogs. It was a success because what happened is he had no ill or adverse reactions from our cells. Patient number 2 was a dog down in Southern California by the name of Portia. This dog, I drove down to Santa Clarita to see the dog, was gurgling. You could hear the lung sounds from across the room. I mean, you could listen to the dog run around, you could listen to him gurgling, and you said, this dog's in trouble. And the dog is in trouble. There's nothing veterinary medicine can do. I can either reach for the blue juice, the euthanasia solution, or I can reach for the red juice, the phenol red containing stem cells. We actually don't use phenol red, but, and I can inject it. Which do you want me to do? It's your dog. What do you want me to do? You want me to euthanize your dog now, or you want me to try stem cells? It's an interesting question because we know how one of them turns out for sure. The other one, we don't know how it's going to turn out, but if it doesn't turn out well, we've always got choice two, or choice, we've always got euthanasia as a choice. And so this owner was very enthusiastic about having us try it. So I said, we tried it right here, patient number 2. First time we treated, slight improvement, not much. Second time we treated it, better improvement. Third time we treated it, I got probably one of the best emails I've ever gotten. And the best email that I ever got from a client, she said, "You won't believe this, but Bedlam has returned to my household." And you'd say, Bedlam? What happened was this dog was the alpha bitch, was the top dog in a 3-dog household. And after the third treatment, to paraphrase Elton John, she let the other dogs know that the alpha bitch was back. You sit over there, you eat when I tell you to eat, you don't go, I'm in charge here. And trust me, Westies are nasty little dogs. Most of them eat veterinarians for lunch. So but they are nasty little, now they're really nice dogs. All dogs are nice. So I'm sitting there thinking, voila, I've arrived. I've got a treatment for a terminal disease, a major disease in humans. By the way, Hoffman LaRoche has paid $8 billion for data that's not as good as this data. So I thought, wow, I've really arrived. This is really cool. So we started treating on treatment 4. I FedExed the cells down there. I called the client about an hour after, just about the time they're doing the treatment, and say, how's it going? She said, Portia's dead. And I went, what? And she said, yeah, Portia's dead. She said, we started infusing the cells. The dog started cratering. Said he was dead in about 15 minutes. And what had happened is this dog had had 3 prior treatments, no response. We've treated over 150 dogs with no response. And I said, what's going on? She actually FedExed me the dead body so I could do a necropsy on it. So I said, what's going on? And we said it was unambiguous. It was my cells. They're flowing into the dog. We had sensitized that dog. And so by sensitizing that dog, and by that way, that's one of the things on how this disease works as a hyperimmune state. So we had produced a hyperimmune state. We infused the antigens into the dog, and you just push the dog right off the cliff. Well, we do necropsies. We stop doing dogs until we figure out what happened, what we changed. And then from then on, we've always do intradermal skin testing. And patient number 4 is another dog. By the way, these dog owners all know each other. Patient number 4 that we've just started has been treated 3 times, has responded very well. On the 4th treatment, we sent cells down. The dog is now sensitive to the cells, so we're now using a different treatment. But patient number 3, which is this dog right here, is very interesting because we said, if they get sensitized to the cells, let's work out something else. So we worked out another treatment protocol, and you can see this dog is now out 100 weeks or about 2 years. Okay, so we're obviously doing something to maintain this dog, and it's working. But this is how fast you can move, how fast you can adapt protocols. Nobody in human medicine has variable protocols. We adjust them as soon as we get feedback from one dog, we adjust them to the other. So that's the advantage of using them. Mechanism of actions of MSCs, there's at least 11 of them. Probably the only one that nobody believes anymore is that they transdifferentiate into specific cell types. That's not particularly thought to be a mechanism by which they work. Growth factor secretion, anti-inflammatories, stabilization of cells, proteolysis exosomes. There's a whole bunch of mechanisms nobody knows. Working them out in a tissue culture dish, working them out in mice probably is not a productive pathway. Working them out in patients, patients is. Okay, so now let's talk about some of the dogs we've treated, although we'll start with a cat first. And this cat has a lipoprotein lipase deficiency. Lipoprotein lipase is a protein, that what it does is when you, I know none of you do it, but when you eat that double bacon, double cheeseburger, double cheese, everything, clog your arteries special, heart attack special. What happens is all that fat gets absorbed in your stomach, goes into your bloodstream in the form of chylomicrons, okay, the little blue things. What happens is it floats around in your bloodstream. What's supposed to happen is there's supposed to be an enzyme, the little yellow spheres, called lipoprotein lipase that breaks these chylomicrons down to free fatty acids where they get absorbed and metabolized in your body. That's what lipoprotein lipase is supposed to do. If you lack lipoprotein lipase, this is two samples of blood. Serum should be, after you spin the red blood cells down, serum should be clear. Should be slightly yellow straw colored. This black lines are an artifact of photographs, but it should be nice and clear. And this is what a lipoprotein lipase deficient person or an animal looks like. Milky white serum, okay? Not good, not good. You get fat, it gets stored all through your body. Well, it turns out that this is a mutation that exists in humans, about 1 in a million. And so there are 60 million cats in the US, and it turns out, so that means there's 60 cats in the US with this lipoprotein lipase deficiency, assuming the same frequency. It turns out 30 of them live in UC Davis. And so this is, they like the double bacon cheeseburgers, what can I say? But what we did is we first checked to see if these are RNA gels or gels for PCR gels looking for expression of lipoprotein lipase by the MSCs, the stem cells. And this is a control. This is one that's been differentiated by adipocytes to adipocytes. And we found that the message expression of LPL was exactly the same, so we just went with control MSCs. We harvested them from an LPL positive cat, put them in culture, took the cells in culture after they'd grown up to sufficient quantity, put them into an LPL minus cat, a cat that had the deficiency with the clogged up serum, with a milky white serum. And then what we did is repeat and then collect samples over time and measured blood chylomicron and measured LPL activity at different time points. And what you see here is these are cuvettes. This is the serum. Over time, we did 2 injections, one right here, injection 1, injection 2, and we pulled blood samples out approximately at these times, indicated by that. And as you can see, this is a pre-injection serum. This is a post-injection 1 serum. Is that clearer than that one? I can tell you I spent lots of time holding it to light, shining flashlights through, trying to do everything to figure out, is this one clearer than that one? And couldn't really tell. Then we did, after a while, we collect another blood sample. Then we did injection 2, and you can clearly see this one's clearer than this one, and this is still clearer than that one, and eventually it gets cloudy again. Okay, this is very exciting, because what this says is this says that these allogeneic cells appear to be clearing the plasma. But the way you check that is, you have your eyeball, the eye test, but the scientific way you do it is dilute it down, read it at an absorption of 600 nanometers of light, and then measure the opacity, and that's what the red curve is right here. And you can see this was a pre-injection sample, post-injection sample, it went down. After a while, it went back up again. We did another injection, it went way down, and then went back up. But we also, in these same samples, simultaneously measured lipoprotein lipase, and you can see that the activity went up. This was huge. This is the first time that I'm aware of that anybody has ever demonstrated that allogeneic stem cells can correct a genetic mutation in a primary animal. It's a relatively trivial problem because there's no orientation, there's no integration. All you have to do is put a cell under the animal's skin, have it secrete LPL, and you will correct this deficiency. Nonetheless, we were very excited about it. Here's over a longer time scale, and this rebound is real. Because what happens with these animals is they process fat so poorly that the body stocks it in the liver, they put it in skeletal muscle, they put it in all the tissues of the body. And so when you put a little LPL in there, what happens is you release more of the fat so that you have a much larger supply of fat, and there's a rebound or an overshoot, so to speak, but then eventually it normalizes out for a long period of time. Well, those are just numbers, okay? Those are just numbers, those are just indications, parameters. But if you looked at the cat Originally, when we treated Alfie, it was the second dumbest cat I've ever worked on. You know, it would just sit there. You go in, you walk into a room, and cats are experts at knowing exactly what you're doing without ever looking at you. This cat didn't even care. This cat just sat there, was absolutely nothing. It was a blob. It sat, and she would sit in the lap and drool and shed. She didn't groom. She was a mess. One of the technicians' job was to groom the cat. Okay, it sounds like a really strange job, but this cat was a mess. After treatment, the cat groomed herself, more complex behavior. Here you can see her after treatment, sniffing around the lab as she's walking, sniffing, kind of curious, gains weight, totally different cat, behavior improvement. This is exciting. But more importantly, like I said, this is the first time that I'm aware of anybody used an allogeneic stem cell. Most people try to genetically correct the cells. We didn't even try to correct the cat cells. We just took a donor cell, put it in the cat, and got it to work. And because they automatically secrete LPL. But this says that you can use a diseased dog can be the human patient's best friend. And so we'll talk about dogs now. Other diseases that we've worked on, and we work on diseases as they come available, depending on where we're at and what's funding and stuff like that, is we did a little bit of work with degenerative myelopathy or human ALS, dilated cardiomyopathy. This is 30 to 50% of Doberman Pinschers will die of this disease. Again, a horrible disease. You build up fluid in the lungs, you can't breathe, you lose exercise tolerance, you lay on the side gasping for breath, and then eventually a vet comes along and euthanizes you. Not a really good outcome to look forward to. This is a dog named Gunner. This dog is about 4 months after treatment. And 4 months after treatment, when 50% of his colleagues would be dead, 95% would be dead in 6 months, this dog is digging 4-foot holes in the backyard. Okay, so he's not feeling any pain. West Highland Whites, I talked about IPF, idiopathic pulmonary fibrosis, but I want to finish up with lysosomal storage diseases. This is a family of diseases, of neurologic diseases, where you have some genetic defect, and what happens is your brain starts building up storages and stockpiles of precipitated inactivated denatured proteins till you start losing neurologic function. And this includes such diseases as Tay-Sachs, Niemann-Pick, diseases, and several other ones we'll talk about briefly. This is a picture of one of these is from, and I don't remember which one's which. One's from a Tibetan Terrier brain and one's from a human child's brain. But you can see the dark pigmented osmophilic granules stored in the neuron that's leading to neuronal dysfunction will eventually lead to death. And I had one person, I'll point this out to you, one person said, one reviewer on one of my grants said, why should NIH pay to fix a problem, a genetic problem that dog breeders should take care of breeding out? Well, I can promise you, all the people in this audience, none of you were selected. Did your parents ever select based on their cardiac genetic profile, their neurologic genetic profile? When your parents met each other, they liked their smile, they liked the way they laughed, they liked something about each other. They didn't select on genetic basis. Most of the people that have this, and you're young married, or will be young married someday, as their thing that they say is, we met, we fell in love, we decided to have children, we had children, and 2 years later we get a call from the doctor that changes our life forever. Okay, this is scary. These are genetic patterns that you can have that can happen to you, and I hope it doesn't. But it does happen about 1 in 100,000 in a lot of the diseases I'll talk about. Includes Tay-Sachs, Niemann-Pick, Batten's disease. There's DNA tests available for them. Unfortunately, most people do them after they have the child, not before. You don't know what your carrier status is. You have no idea what your carrier, I have no idea what my carrier status is. So there's no reason you should know that, but it is something the next generation will have to deal with. The diseases we've worked on is cathepsin D deficiency and an ATP13A2 deficiency. That's a protein none of you and I had never heard of it before, but it goes by the name of the early onset Parkinson's disease, or the PARC9 gene is what it's called. This is a disease that's a very rare form of Parkinsonism, and this is a Fluorodopa study of Parkinson's disease. And this is what a normal brain should look like. This is the striatum. The bright red those are the areas where dopamine, many of you have grandparents who have Parkinson's disease, or you've seen Parkinson's, you know what it is. This is what a normal brain should look like with bright red. That's where the dopamine is stored and working in the brain to prevent the dyskinesias of Parkinsonism. These are 3 human patients that all have early onset Parkinson's disease. You can see there's no red there. Turns out Tibetan Terriers have the same genetic disease. They have a deficiency in dopamine. We've never done a fluoro scan on them, but this is a dog. This is a human patient, age 16. He's got motor dyskinesias, age 40. He's just about ready to die. So he's had 24 years of immobilization, of tragedy to deal with. This is a dog named Boots. This is Boots, and let me see this is Boots here, and what I'm going to have to do, and I usually do it, and I apologize, most people embed films in there, but if I embed films in there, what happens is you usually get screwed up. So we'll play Boots. This is Boots, the dog, before treatment. This dog would, and I would have put this dog down much before this, lived in the client's closet. They were scared to come out, would seizure 5 or 6 times a day, would fall downstairs. Okay, this to me would be a candidate for euthanasia. This is what it looked like before treatment, just not a very good happy camper. And this is after treatment. Still not a perfect dog. Still hitches and get along. You'll see when he turns around here, he'll lose a little bit of back end stability. But nonetheless, an acceptable pet. See, there's a little bit hiccup there. And so that is what you deal with in human medicine. So what do? Do you put the dog down, or do you try something new and different? Well, fortunately, these people, we had already treated one dog that was friends of these that had this same dog, and they told me about her, and I'd actually originally passed on treating Boots because it was so severely affected. I thought a false negative would be worse than not treating, so I'd passed on the dog. And then I called up after we'd had success treating one dog. I called them and said, "Is that dog still alive?" And I'll never forget this. I'm driving across the middle of the night, driving across Ohio, coming back from a dog show. And she said, the person I was talking to who knew these people said, "I don't know, I'll call them and give you a call back." And she called me, and this is where Boots was. You know, when the video falling over and having seizures and all that stuff. And I called them up the next day and said, "Can you give me 2 weeks?" And they said, "Yeah, we'll give you 2 weeks," because they didn't want to put their dog down. So I flew to Toronto, treated the dog here, here, and here. And as you can see, it almost became normal. It certainly wasn't normal. I don't want to pretend that that was a normal dog, but this is the second try on a second patient. I don't expect to have everything worked out at that point. So we treated the dog 3 times. The dog became, I would call it acceptable as a pet. And I'm thrilled. I get really excited about this stuff, and I was absolutely thrilled. And then, I never forget this one either. I'm in Philadelphia, and I get a call right about here, and they say, "Boots has relapsed. We're going to put her down." And I used every persuasive technique I could do. Say, "Please don't, please don't. We're in the middle of an experiment. We can work it out. We can figure something out." So what I did, I'm in Philadelphia. Pamela and I drive all night from Philadelphia to Buffalo. We have cells FedExed into Buffalo, pick them up at 9 o'clock in the morning, drive into Canada to treat the dog here, here, and here, and you can see the dog got better again. Okay? This was very exciting because now, again, you're reversing a genetic condition by using donor cells from a donor dog. After here, the dog was eventually succumbed to treat in spite of treating, and what I believe is happening here is these are foreign cells. They're not tissue matched, they're not patient matched, and probably with humans, if you did better tissue matching, it would not be a problem, but it certainly was not something we can do in animals, because that's about $10,000 worth of science right there, so you really can't do that. But it was very good. And this is this is confirmatory PCR showing it expressed the correct MSCs express the correct form of ATP13A2. This is a control brain. That's a red blood cell. This is that's a red vessel, a vein, and this is showing the endogenous fluorescence from the hemoglobin. That's just a control. There's nothing there. This is Boots's brain right here. You can see the fluorescence, the red blood cell, the vessel here, the vessel here. All of these lesions here are the precipitated ATP13A2, the protein that's mutated. This is what's causing that dog to die. That's what killed that dog, is that right there. Apparently the cells did something where they proteolyzed it, stabilized neurons. We don't really know at this point. And we've treated 8 dogs. I'm pretty comfortable that this is working now. But more importantly, what's going on with this is a table of CNS diseases, storage diseases of dogs and cats. This is a list of the human diseases. Okay, Batten's disease, Tay-Sachs, Niemann-Pick, Gaucher's, Krabbe's disease, and GM1 gangliosidosis. These are all diseases that you can have as a parent of a child that you worry about. These are all terminal diseases. Your child will be dead by the time he's 12. Probably less than that, probably by the time they're 8. Is there nothing we can do for these diseases? The ones that are in green, doesn't look very green to me, but in green are the ones we've treated. This is the early onset Parkinson's, the ATP13A2. They expressed a correct copy of the message. Cathepsin D, American bulldogs have this same disease, and yes, they express the message. But look at all these other places where the MSCs express the correct copy, the correct form of the message. This has all been done with PCR, measuring the mRNA in the cells. So in theory, each one of these cells could be used to treat these animal patients. Big Pharma will not go into these diseases, and Big Pharma won't go into them because they're 1 in 200,000 diseases, and there's just not enough money for them. And this is one, GM1 gangliosidosis. I just got this off the internet. Basically, they're once per 100,000, 200,000. Most children die about age 2. Okay. Is there a dog solution for that? Well, this is gangliosidosis in a Shiba Inu. A Shiba Inus, this is when they're born, they're normal puppies. 4 months, they're still pretty normal. 6, they start having ataxias, dyskinesias. They're starting to seizure, lose it. They start seizuring here. By 13 months, you usually put them down about a year. And so, can we treat these dogs? What's it take to treat these dogs? All it takes is some cells, hit an IV, and infuse them in the cells. So you can really learn a lot from these models. If you want to call them models, I call them patients. But there's a lot that we can learn from this. And basically what I'm advocating is that basic science, you still need basic science. You need laboratory animal testing, but once I get laboratory animal testing where there's a good example of laboratories supporting the use of the justification of moving these into animals, I'll put them in animals, and then this information can be used to, one, get approval in animals if you want, but also more importantly, get approval in humans. And which is where let's face it, all of us do research for improve animal care, or human care. And so what happens is, can we go from here to almost a phase 1? And the reality is that FDA, on many of those childhood diseases where there is no treatment available, there's prevailing evidence in a closely related model, and there's no other treatment, they will grant what's called compassionate use approval. So literally, you could take the Shiba Inus, do 6 or 12 or a dozen of them, and if you get the data that I expect you would get, you would be able to take that, go to FDA, and have approval to try it in one child. Very interesting. Instead of 800 million to get from here to here, you can do it very quickly. And by the way, clinical data always trumps preclinical data. And this would be called clinical data because it's a single copy mutation in a naturally occurring disease. So, okay, with that, let's lighten it up a little bit because that's too heavy. I'll give you a few of my thoughts on dogs. These are my personal thoughts, I might add. Dogs ask so little, they give us so much. You will never get the unqualified love that you get from your dog from a person. And that's certainly not from your spouse. Everything from a spouse when I was married is negotiate. Honey, you take out the garbage, I'll mow the lawn. You clean the garage, I'll make the bed. I'll do the laundry. Everything's negotiated in a marriage. Your dog just says, can of dog food and I love you. I mean, what could be better than that? Anybody who doesn't know what soap tastes like has never washed a dog. And the most affectionate creature in the world is a wet dog. You ever notice that? They jump on your bed when they're soaking wet, so they love to jump on the bed. This is the one I like the best. I wonder if other dogs think poodles are members of a weird religious cult. So I think they are. So, okay, the last one. This was in the B about a month ago, and this is for all those people. So this is the express lane. And ascending into heaven, and here's the wife again saying, "What do you have to say now, Mr. Nobody is looking?" for the people who didn't clean up after their dogs. So, with that, I'm willing to answer any questions. But we shared a little philosophy here. So. Thank you. What?
Do you know any pet insurance covers?
No, pet insurance will not cover any of this. I'm sure of that. They don't generally do experimental treatments per se, and this certainly would fall under that category. But what we try to do is find granting agencies that will pay for it is the goal, and we try to treat for free and find granting agency that will pay for the materials. Any other questions? Is that clear? Oh.
Actually, kind of like mechanistic questions.
Okay.
For your cat model, were those feline bone marrow allogeneic cells, were those from pathogen-free cats?
Actually, they were. Yeah, the reason they were pathogen-free is they came from the same colony, and that's an SPF colony that we have at UCD.
And then my second question is, when you're injecting stem cells, are you injecting frozen cells reconstituted, or are they fresh from culture?
Fresh from culture. We harvest, and yeah, we don't do frozen cells. I'm not sure. Have you done those? They don't particularly like the frozen cells. I'm sorry?
That stem does it, I think?
Yeah, I'm not real wild about that. But they got that from humans, and I'm not real wild about it in humans either. There's another question over there. All the way in back.
Where do you get how do you get the patients?
Actually, what's happened is most of the diseases we're working with are very rare diseases. And so what we do is I'm actually pretty active in the American Kennel Club, and that when they're breed specific, I can go to that breed club, put out a letter in their newsletter or announcement in their newsletter or something like that. Or actually what happens is they all know somebody who had that dog that had that disease, and then they contact me is usually what happens. But there is a whole network of the breed specific diseases. Yeah, the question is, when I'm injecting, are we injecting into the blood? We have only a long time ago, we used to do tissue injections where we were injecting into the brain and injecting into tissues. And what I found is, although the cells are supposed to migrate, in my hands where I checked, what you wound up with, and I think scientists are not as honest as they should be. Bite my tongue, dishonest scientists, but we'd inject maybe 100,000 cells into it, like I am, or in the brain or something like that. And what we'd see is maybe 10 cells that would migrate away. And so I'd report, these cells migrate through the brain. I didn't do that, but report these cells migrate through the brain. The reality is 99% of your cells are stuck there in a glob of stem cells and actually forming a scar tissue or a granule there that really shouldn't be there. So what we have done is all of our injections have been IV, basically through the cephalic catheter on the forelimb. Question?
Do you have any notable results on any control?
I'm sorry?
Do you have any notable results on control groups?
We haven't done control groups when you say that. You do control groups basically for safety testing because you don't have anything to measure. If you do a dog that has heart failure, for example, you can change the parameters in the cardiac, like the fractional shortening goes up and stuff like that. In a control dog, it wouldn't change. It would just be that. We basically did that, and that was why we did the degenerative myelopathy dogs, because those are normal control dogs. Their back legs don't work well. And so they have normal kidneys, normal heart, normal lungs, normal everything, except their back legs don't work so well. We don't like to kill dogs. I mean, that's one of the issues that we had, is there was an argument, well, you shouldn't be injecting patient dying dogs. You should be injecting control dogs, and they should chop them into little pieces. Well, to me, if you know anything about animal rights activities, probably 25 years ago, one of the PETA people said, well, instead of, what we should do for controls and biomedical research to test new drugs is we should use people on welfare and people in prisons. And that's the same thing I hear when you say, take a normal dog and inject it and then kill it. I don't like to kill dogs, and I won't for that reason. And that's why I started doing DM dogs, because the degenerative myelopathy is basically a healthy dog. And the other problem you have is you start buying dogs from suppliers. The whole concept of control is a very interesting concept. So I go buy 2-year-old dogs from Charles River, and now I'm treating your 11-year-old German Shepherd with degenerative myelopathy, or your 8-year-old Doberman Pinscher with heart failure. Is the 2-year-old Beagle I bought from Charles River a control for your Doberman Pinscher? I don't believe so. It is a dog. It has 4 legs. It's got a tongue and it barks, but I don't think that's a control for it. And so it's very interesting when you start looking at controls. But that's a very important question. You need to know what you're comparing it with. Fortunately for us, most of the diseases we do are one-way streets. The IPF dog is not going to get better. They're going to go until they slowly get bad, or until they die. But it does need to be done. To do a study with controls like that's probably $200,000 to do it right. Same thing with the, in fact, you don't even have that number of dogs to do the Parkinson's disease, because I couldn't do it. But you do controls for safety primarily, or initially. Question?
What do you think is the actual mechanism of action that is alleviating either their normal phenotypes or seeing?
I'm sorry, inject them into where, intravenously? Okay, what I believe going on, and this is actually very interesting, is I believe the reason you're younger than me, isn't because you were born a lot later than I was. I believe the reason you're young, at least part of the reason you're younger than me, is because your stem cells are in a lot better shape than my stem cells. Because I'm old and my stem cells are 1 in 500,000, yours are probably 1 in 50,000. It's a very interesting concept if you carry that out biologically. What is aging? What is this kind of thing? But it is clearly a cell type that is depleted with aging. And I believe the cells, I don't believe the stem cells, I call them stem cells, I don't believe they're stem cells at all, I believe they're reparative cells. And I think that that's part of, they repair life's normal wear and tear. And I don't think that they necessarily, I don't think they go to my brain and give me new neurons, for example. I think that I've got old fossilized neurons and they help them function a little better, kind of thing. At least that's, but there's no evidence for any of this. This is obviously a speculation. We haven't done that because, again, these are client-owned animals and we can't really, that's one of the downsides of what I'm doing. The reality is it's good that I'm taking real diseases and treating. The downside is I don't get to call the client up and say, well, it's time to chop your dog into little pieces. It's their dog. On the other hand, there are modern imaging techniques that you can assess some of those things. If we had more money and more support, we'd probably do that. Question?
Stem cells are, you're injecting the stem cells and they're producing enzymes that are lacking in these dogs that are causing them to get diseases. Does the stem cell then somehow, do you see an increased gene expression just of those particular enzymes that are required, that there's somehow some type of communication going on? Or are they producing all types of enzymes and it's just that now you have an increase of the one
That's an excellent question. The first part is yes. Second part is we don't know. When I say that with the lipoprotein lipase, where we actually measured the enzyme, I'm pretty sure that came from the stem cells. And so they did go in and synthesize that enzyme. Do they have a way of sensing what they need to do? One of the things that's very interesting, and I'll just share it with you, is most of us don't do a lot of quantitation in stem cell field. If you say, let's say even in my old decrepit state, I have 10 billion MSCs floating in my bone marrow, why do you think injecting 1 million cells would make a bit of difference in that respect? And so these cells seem to function in some manner, shape, or form as, to me, they're reparative cells more than they are regenerative per se. But that's a very important question to ask. It's an even more important question to answer. And unfortunately, I can't.
Has anyone looked at genome-wide expression between dogs and humans under stress, like folks do with the mouse?
I don't think they have. The dog is not very popular research model anymore, and a lot of that is PETA. And in some respects, I admire PETA because I do think that historically biomedical researchers have stayed away from dogs because people get emotionally the way it plays in the newspaper. Everybody's seen a picture of a cat with electrodes sticking out his brain. The idea, in fact, I wrote a grant to Doris Duke, and they wrote back to me, turned it down, and they said, Doris Duke Foundation does not support animal studies. And as soon as I got that letter, I called them up and said, wait a minute, if you know Doris Duke was quite an animal person. She had 6 horses, about 12 dogs. She loved animals. And I called the head of the foundation, or the manager, whatever it was, and I said, look, this isn't research on animals, this is research for animals. Doris Duke would approve of what I was doing if she were alive to see it. And he just wouldn't budge. He's a bureaucrat from the word go. No, we don't do animal studies. A lot of the reasons they don't is because they're so worried about PETA that PETA will, I don't know what they're worried about PETA doing. But the reality is, everything we've done is for the betterment of dogs. And it's a very interesting thing. But the idea, like I said, what are the alternatives? The alternatives are do mouse studies and then go right into humans, or find a dog with a net, or cat or horse. I'm not a speciesist that it has to be a dog. I mean, to find a naturally occurring disease that closely parallels the human disease and then test it in that and then go into humans. But instead, our government will say, no, we have to wait 15 years and pay $800 million. If I had a child with any of those diseases on that table, and you'd shown me data from 6 dogs that it worked, I'd try it in my child because I'd be desperate, and I'd want to try it. And you'll find that a lot of parents of those same groups feel exactly the same, at least from the ones I've talked to. And so then you got FDA saying, no, we can't let you do it. We're going to let your kid die. That doesn't cut it with me, and I don't know how. So we as a society have to work out that moderation. We can't have crazy mad scientists running around. On the other hand, we can't deny therapy to those who need it, and if it works. And by the way, one other thing I forgot to tell you guys. There's a big question whether stem cells work in any diseases where it's been shown to demonstrate to really work. And probably the best example of this is, I don't know how many of you know this, Peyton Manning took a year off and he injured his neck, ligament of his neck, and went over to Europe and had stem cells injected in his neck. Now you can interpret that data, this sounds like two picky scientists, you can interpret that data 2 ways. You can say, well, Peyton Manning had stem cells in his neck, it must work because he's still playing football. Or you can say he lost, therefore it didn't work on the Super Bowl. I'll be up here, and if you want to ask questions, I'll be more than pleased to. But I think you guys got to go. Where are we at the 7th inning? She's supposed to. Yeah. Anyway, so you can announce what the score is if you want. So anyway, thank you all. Been a great audience.